Yes, my name is Russell. And as Molly mentioned, I started out as a mathematics undergraduate. Um, so transitioning into botany has been, it's been a lot of fun. It's been much more application based. And actually, learning how I can use mathematics to sort of do something in the world. So my master's project is called TSS-Pro. It's a plant promoter architecture prediction tool. It's a computational tool. Let's just jump right into it. So essentially, to put this all in context, what are we talking about biologically? Well, the project that I'm going to be working on is all about transcription. And essentially, to just get us all on the same page, we're mostly worried about how transcription is influenced by regulatory elements called transcription factors that are needed to recruit the Pol-2 complex, the RNA polymerase II complex, to a strand of DNA to actually initiate transcription. So how do we do this? How do we actually turn this biological concept into computational tools, numbers, data? Well, we use transcription start site sequencing, which at a high level takes the 5' end of mRNA transcripts, sequences them, maps them to the genome, and then once they're mapped to the genome, this collection of TSS-seq reads, we find it runs into these clusters of reads that show where these transcripts have started. And so we take a particular cluster that is generally upstream of a gene of interest, some annotated gene, and we take just the statistical mode of the start sites for this collection of sequence reads and call that just the TSS, the transcription start site, just to use a single location so that we actually have one point that we're sort of concerned with. So where does this project, this tool, actually come in? Well, it's a software tool to help us identify functional groups of these cis-regulatory elements using transcription start sites. So by using this TSS-seq data, we can hopefully find these functional relationships amongst these transcription factors that recruit the Pol-2 complex that's necessary for transcription. So what can you do with this tool? What's sort of the end result of this project? Well, if you have high resolution TSS-seq data, you can discover particular cis-regulatory elements that might be influential in regulating particular genes or genes of interest, which you can use in pathway engineering or pathway discovery or promoter engineering to discover these groups of elements that you might need or want to use in a promoter to promote transcription of a particular gene. And if you don't have data, if you might maybe have not enough data, or you just don't have an organism particularly that has a annotated genome, you can possibly apply a model from one sampled setting, such as developmental time, tissue, environmental state, et cetera, in a particular organism, and then try to infer the promoter architecture, these groups of regulatory elements, to a different setting, perhaps from one developmental time to another, or one tissue to another, and so on. So how do we actually do this? How does the tool actually accomplish this? Well, again, the main question that we were sort of asking is, for a given gene or a set of genes, for which we have a transcription start site sequence data present, what are the regulatory elements that actually, or possibly, contribute to the transcription in this data set? We know that there's sort of possible combinations of these transcription factors, and we're hoping to uncover what these combinations actually might be. So where do we look for regulatory elements in the genome if we're just given an organism's annotated genome, where do we start looking? Well, one logical place would be in promoters. But the sort of textbook definition isn't essentially distinct enough. We don't get just a single set region that we're supposed to look in for promoter regions that tells us where to look, but it's a starting point. It reduces sort of a search for regulatory elements that might be influential from the entire genome somewhere to at least a location that might be associated with transcription. But this is the sort of the first problem with this approach, is that where are exactly our promoter regions? Where is this really defined to be? There's not typically a sort of annotated region for a gene that says, this is the only place that you're going to find regulatory elements for this gene, and don't look anywhere else. You kind of have to figure it out on your own. So we turn to a computational method to come up with regions of enrichment for particular regulatory elements. And what this essentially does is it takes, for instance, the TATA promoter element, and we ask, where might this be influential in relation to a particular TSS? And so we can take the individual seq reads in the TSS-seq dataset and look at particular elements and find these regions where there seems to be much greater activity for those particular elements than, sort of related to another region. And as, an example, we see that the TATA region of enrichment turns out to be, from the data, roughly 30 or 40 nucleotides upstream of transcription start sites. For other regulatory elements, it might be, like in this example, 40 nucleotides upstream of a transcription start site. It depends on the regulatory element. And so trying to just search within promoters for anything isn't really distinct enough. We want to switch it so that it's more specific to the regulatory element that we're looking at for each element, so we only look in particular regions that actually matter for that element. But a second challenge comes up with this, which is, how do we actually quantify whether a regulatory element is present or not, if it's exerting some sort of influence? Well, we use a sort of computational method called a positional weight matrix. And at a high level, it's a way to model a particular sequence and the likelihood that that sequence is, or a regulatory element, and how likely that regulatory element is to bind to sequence. And so in this image, you can see that if you line up these letters, they're not very similar. And so you receive this low score from scanning a positional weight matrix. Whereas if you scan a region that is very similar to the positional weight matrix, you receive a very high score, indicating that in this region with a high score, there's a much higher chance that a regulatory element represented by this sequence is going to bind in that location. So how do we actually put these pieces together into a model to use within the tool? Well, the output of the PWM scanning is a likelihood score that represents the likelihood of a functional regulatory element actually being present in a given sequence. And then in a particular data set, we can use those scanning scores as features within a machine learning model, generally speaking, to come up with some sort of relative importance of these regulatory elements in the particular data set. So that once we've run a particular method, after we try and learn this relative importance, we can come up with sorts of answers to questions that might ask, what's the probability of something happening given this particular TSS-seq data, or some sort of data representing transcription start sites. And this is sort of, it's left as a blank because the particular machine learning method is kind of dependent on what question you're asking. For instance, if you're simply asking a yes or no question as to whether a particular gene is of a particular type, maybe you're asking if it belongs to a particular family of genes, you can use a specific method that is suited to that kind of question. It sort of depends on the context. And so it's really left open because within the tool, you'll be able to sort of tweak how you actually want that. And so once you run this sort of whatever method that you've chosen to run, you get this model output that allows you to answer these sorts of probability type questions. And you can then start to investigate what's given by the model. You can ask, what might be these highly relevant regulatory elements for particular functional binding sites? And you might say, oh, I want to find binding sites that are related to what I find in my data set. That's one of the sort of most obvious use cases for this tool. And you can also, as I mentioned earlier, apply the model to a situation that you might not have the particular information or the particular data that you really are hoping to have. And so in addition to this sort of slightly open-ended tool for transcription start sites, you can also apply it in a more general sense, hopefully, of course that's the hope, to other sorts of ideas. In one case, we chose to look at genome mining of trying to find genes that were belonging to secondary metabolism, just as a sort of test case, you know, a use case to sort of throw this out and see if it sort of worked in general. And we tried to create a model to classify secondary metabolism or genes that are related to secondary metabolism and ask, if we run this on, if we include every gene in the Arabidopsis genome, what is going to be classified as a gene that might be related to secondary metabolism? And it turns out that there's a lot of tweaking that should be done, but it's possible to do. And the results might not be perfect due to sort of different limitations, but it is a sort of feasible use case. But it sort of brings up that there are these limitations, that it depends on the data that you're using as to what sorts of questions you can ask and how you can sort of ask those questions. But the goal is that the tool is open enough and sort of generally useful enough to allow different sorts of questions to be asked. With that being said, does anybody in the audience have questions?
If you have a question, just, all right, John Fowler.
Hi, Russ. Can you hear me?
Yes, I can.
Thanks for the talk. So, so I have a question that's a little bit related to taking your successful model, and I'm going to call it successful model, although I understand that's the goal. You're not at that point yet.
Right.
But let's say you have this successful model, and my understanding from the talk is this is probably going to be sort of tuned into Arabidopsis. Is that correct? At least as a first guess.
It depends. It's purely dependent on the data that you use. If the data that you use is from a particular organism where you've mapped it to that organism's annotated genome, then it would purely be tuned to that organism.
Okay, so you know, thinking about regulatory element architecture, it sort of depends on the type of transcription factor that's going to bind there, and I can sort of hypothesize that there might be two sort of general classes of transcription factors. One in which the binding needs to be a particular positional distance from from the transcription start site, and another where the transcription factor has a little bit more flexibility, can sort of be in multiple places, or relative to that transcription start site.
Right.
And Arabidopsis has a really small genome, and one of the reasons it has a lot fewer transposable elements. But in larger plant genomes, you have the possibility of having sort of DNA insertions that might move CRE relative to the transcription start site.
Right.
So sort of as a hypothetical use case, could you sort of use your model to sort of look for CREs that sort of are more constrained in terms of their position relative to the TSS versus less constrained, right? In other genomes, maybe these particular CREs can move about a bit. Does that
You
make?
could. Yeah, I think I understand what you're asking in the sense of, are you asking if you could constrain it further or sort of allow it to be more general?
Yeah, I mean, I guess the question would sort of be, if you do train it on sort of multiple genomes, different genomes, could it sort of help you recognize particular CREs that are more constrained or are less constrained relative to the TSS?
Oh yeah, it definitely could. There's different ways you could use the tool to do that, but essentially, yeah, you'd be able to sort of look within these regions of enrichment and find if there's sort of consistency, and you could look within the results and sort of see that, oh, in some cases, certain regulatory elements are consistently having a single sort of region of enrichment, whereas others not so much. You might have multiple distinct locations, like you're saying, but you'd definitely be able to do that, to look at those sorts of different cases where certain elements might have multiple locations, whereas others are much more specific.
Okay, cool. Thank you.
Yeah, thank you.
I have a question, if you don't mind.
Yes. No, of course.
So Russ, the output for your model, is it just whether that gene is expressed or not, or is there any sort of future plans for incorporating sort of the level of expression with the presence of different CREs or classified as CRE associated?
It could be, and there's actually another project that does include, just within Molly's lab, that does include sort of using different features, essentially, because for this particular tool, it's, as you were saying, it is mostly looking at presence and sort of abundance or affinity for particular regulatory elements, but you could tweak it to include those sorts of other types of data, like open chromatin or gene expression. You could most definitely just sort of tack that on within the model, or sort of within the tool. It would be fairly straightforward to allow it to include other types of data, to sort of put those all together to try and use those as the sorts of features that are attempting to be learned as to their importance to the question that you're asking, with respect to that data set, if that answers your question.
Yeah. Sounds great.
Awesome.
Bruce, you have a question?
Yeah, I have a question from out in left field. Sorry about this, but I'm intrigued by your background as a linguist and coming into reading nucleic acid language, and whether or not there's any similarities or differences there. In particular, we think about learning something like English or foreign languages, and they're quite complex. There's illogical things about them. There's redundancies in them. and we're using data mining tools on natural language as well. So I'm, as a linguist, how does DNA compare to, let's say, English?
It's actually a really interesting question. A few fall term, and actually last term, I took 2 classes that kind of opened my eyes actually to these, like you're saying, different data mining techniques to biological data and specifically DNA data, and how similar they are in the sense that in a lot of text mining applications, you have a similar sort of structure in the sense that you have a string, a sort of linear sequence of words or sentences or letters, even, depending on how you want to sort of look at it in terms of the resolution. And methods that can be used for text mining are almost directly applicable to even this particular use case of looking at transcription factors, in the sense that you have these sort of discrete units that just sort of show up in particular combinations. And essentially the question at the end of the day is you're trying to ask, as you are in language, you're trying to say a sentence is a collection of words. So what does this collection of words mean? Maybe it's some sort of more theoretical level. You're kind of trying to ask the same question in this case of either transcription factors or maybe other types of elements in the sequence of DNA, that you're trying to pull out these discrete units and compare combinations or connections between these discrete units and trying to get some sort of theory out of it that asks, what's the sort of higher level meaning of these combinations of discrete units? And there's all sorts of really cool applications that you can use with this, because these 2 things are so connected, that it's definitely been a really sort of exciting realization of my own. It's like how many cool different ways you can do this sort of data analysis on DNA. And yeah, it definitely really follows certain aspects of written language.
Great, thank you.
Yeah, no, thank you. That was a cool question.
So Russ, I have a question. This is Joey. When you're looking at these, transcriptions, the transcript factor recognition sites,
Right.
and I guess, again, this is not my area either, but it seems like you're going to have some sites that are more similar in sequence than others.
Right.
So you may have 2 transcription recognition sites that are very different, and some that are very similar. And those that are very similar, it seems that you could have minor mutations that might shift the recognition site to different transcription factors.
Right.
Can that be modeled, and does that kind of provide any insight into like the number of regulatory networks or common regulatory networks are possible, and the shifting between networks? Is it possible to have one gene that at one point is part of one regulatory network and then due to mutation shifts to another?
A little bit. In terms of like the idea, it's definitely possible. You sort of wrapped up in the idea of this positional weight matrix scanning would sort of quantify that idea a little bit, in the sense that you'd have these sorts of signals that just kind of, meander about, and you'd have a sort of, point that might say, here it matches up really well, but as you're sort of saying, if you shift it a little bit, if there's a tiny, only a few nucleotides different from maybe some mutations or something, the signal, the sort of score signal here might be in a certain range. You could use that as a sort of numerical feature to categorize these sorts of the differences in these sequences. But I haven't really thought about that much. But it definitely, in this sort of, the idea of these sorts of scans, you could definitely quantify those differences a bit to try and categorize these things to possibly come up with those types of networks that you're saying.
Any other questions for Russ? All right, let's try something here. Everybody unmute and let's give Russ a clap or two, and then we'll mute back. All right.
Nice job, Russ.
Nice job.
Great job.